Lastly, in restriction enzymes, we come about with relation to knowing about what are the applications of restriction enzymes. Restriction enzymes are basic tools in genetic engineering or recombinant DNA technology. By using restriction enzymes, what is it that human race or the advances in genetics and biotechnology can derive? Firstly, from the source DNA which is isolated, which could be either a plant DNA or an animal DNA or a microbial DNA or a human DNA, we can cut the DNA into smaller fragments. By using restriction enzymes, we can cut the DNA into smaller fragments. After cutting or chipping the sort of a DNA into smaller fragments of variable lengths, which is recognized by specific sort of a battery of restriction enzymes, you can easily isolate the gene of interest. There could be a gene for flowering, a gene for fruiting, a gene for photosynthesis, a gene for nitrogen fixation, and a gene for early maturity. Thereby, we can isolate the specific sort of a gene of interest. So, this method is one very important application of restriction enzymes is we are moving into precision molecular biology or precision engineering, wherein all unwanted genes can easily be taken off. So, the second important application is unwanted sequences can easily be removed. That is, we can cleave out and remove off all the sort of an unwanted DNA. So, all the junk DNA or the waste DNA or repetitive DNA or non-functional DNA can easily be eliminated from the from the source DNA, and it is very specific in its sort of an application. The third important application which has got a lot of relevance to plants is we can construct a restriction map of the DNA of the species. If you just take the case of rice or maize, in case of rice, if you can isolate the DNA, which is a very long molecule, we can cut the DNA of rice can easily be chipped into multiple pieces, and the multiple pieces are just like a sort of a stack of pins or just like a stack of needles, and we can, exactly knowing which enzyme can cut the sequence length, we can align the sort of a sequences in a particular pattern and develop a sort of a restriction map. A restriction map will exactly know where is the gene of interest, how many nitrogen bases are present in the gene of interest, what is the distance between one gene to another gene on the sort of a DNA of the organism. Thereby, a restriction map is just like how do you find the sort of a direction between one bus stop to the other bus stop, and what is the distance between one bus to the other bus. And thereby you can know the distance between the destination from one place to the other place. Similarly, when you use the same analogy in molecules, if you take a DNA molecule, we can know what is the distance between, say, nitrogen, between the sort of a nucleotide number 1 and nucleotide number 2. What is the distance between first nucleotide and the 100th nucleotide? Thereby, we can develop the restriction map of DNA. Restriction map of DNA has been done in the case of rice, maize, wheat, potato, and even with relation to brinjal and even tomato. Most of the sort of an organisms which are primarily used for as genetically modified foods. A very important application is when you use restriction enzymes, it is a source of restriction fragment length polymorphism. That is, you are using restriction enzymes, you are developing small fragments of the DNA of variable size, and you are of variable lengths, and because they show variable lengths and variable sizes, they show polymorphism. So, RFLP, that is restriction fragment length polymorphism, is a very important attribute or a character of different species. An easy way, how do you primarily know about RFLP is, it is used as a sort of a tool or an aid for DNA fingerprinting. In an event that you want to exactly characterize or know what are the properties of rice, or rice, what is the origin of rice and which is the true basmati rice. We understand that basmati rice could easily be developed in Punjab. However, we do not know the exact origin of the basmati rice. Rice in entire Asia has originated in Cuttack, and it is in Bhubaneswar. We have also South Indian rice. We also have North Indian rice. We have Philippine rice. Again, rice called Texan basmati rice is a sort of a rice strain which has been developed in America. And if you take countries like Pakistan and Afghanistan and Bangladesh, even they claim to be the places where there is a source of rice. So, in case you are a molecular biologist and you are interested in genetic engineering and biotechnology, we can very easily isolate the DNA strands from all these particular species. That is, we have the South Indian species, we have the Northeast rice species, we have rice species from Philippines, Bangladesh, Pakistan and America. Taking all the rice strains, if we cut the sort of a DNA by using restriction enzymes, we can get a sort of a fragment, we can get a sequence which is termed as restriction fragment length polymorphism. And based on the restriction fragment length polymorphism, we can easily deduce the place of origin of rice, what are the sort of a native genes which are present in rice, and exactly claim about the sort of a legitimate origin of rice and how rice belongs to one particular sort of a country, which is termed as gene patenting. Thereby, the first step towards gene patenting, RFLP, restriction fragment, restriction map of DNA and getting to know the gene of interest is all primarily based on the first tool in plant biology, on plant molecular biology, which is termed as restriction enzymes. While studying on the tools in biotechnology, the second tool is termed as a cloning vector. We had initially read about restriction enzymes. Restriction enzymes are specific enzymes which can cleave the DNA. Now, once the DNA is cleaved and the gene of interest is isolated, the gene of interest need to be put into another sort of a molecule, and it should be transferred from one bacteria to another bacteria. And that's one of the places wherein we use a sort of a vector. A vector is the term which is used for a sort of a DNA which moves from one bacteria to another bacteria. And the vector again is termed as a sort of a cloning vector. The reason it is termed as a sort of a cloning vector is it has got the sort of a recombinant DNA, and the recombinant DNA has to have the properties of making multiple copies of the gene of interest. So, we need to make multiple copies of the gene of interest and load it into different sort of a bacterial species. That is one of the reasons why it is termed as a sort of a cloning vector. Now, when you take a particular sort of a cloning vector is nothing but a sort of a DNA molecule. It's a sort of a DNA molecule which is extra chromosomal. Like if you take a sort of a bacteria, any bacterial cell will have its own bacterial DNA. Now, along with the bacterial DNA, there is always the presence of an extra chromosomal element which is a circular molecule which is outside the chromosome. So, it's called an extra chromosomal element, and with this is termed as a sort of a plasmid. So, what exactly is a plasmid? A plasmid is an extra chromosomal circular DNA molecule which is present just like a sort of a resident doctor. It is present within the sort of an E. coli cell, and the plasmid primarily is inheritable. That is, it can be transferred from one generation to the other generation. Now, when you take a particular sort of a plasmid, what are the characters of the plasmid? First thing is, the plasmid is the cloning vector. The plasmid is the cloning vector, and the plasmid primarily is a carrier of the DNA. It is a carrier of the DNA, and it can make multiple copies of your gene of interest. If you have a gene of interest, like you think you have a gene of interest of flowering, your gene of interest of flowering can be loaded into the plasmid. It can be loaded into the plasmid, and the plasmid keeps on replicating along with the bacteria, and thereby your gene will also be moving along with the plasmid, which is just like a mother piggybacking a child. So, your gene of interest will primarily get loaded into the plasmid, and the plasmid primarily will move from one bacteria to the other bacteria. So, a plasmid is a carrier of the DNA, and it is capable of making multiple copies of the gene. Because when you take a particular plasmid, a plasmid has got a few characters. Now, what are the reasons why you call a sort of plasmid as a sort of a cloning vehicle? It has got specific sort of a properties, or we have some of the sort of properties of plasmids, or what are the properties of a good cloning vector? The properties of a good cloning vector or a plasmid are as follows. First thing is, it should be easy to isolate. The plasmid should be easy, it should be very easy to isolate. If you want to remove a plasmid from a bacteria, it should be easy to isolate. Next, it should be, we should have, it should carry the gene of interest. It should carry the gene of interest, and it can also be reintroduced into the bacteria. So, you isolate a sort of a smaller circle, the extra chromosomal element. It is just like a sort of a bangle, and you can cut the bangle, put your sort of a gene of interest into the sort of bangle, and again take the bangle and load it into again back into the sort of a bacteria. So, you will be able to reintroduce the gene of interest. And when you take a plasmid, the plasmid can occur naturally in the bacteria. It is normally present in every bacterial cell. So, it is a big sort of a boon, or plasmids are a boon in biotechnology because of their natural occurrence, and they are extra chromosomal, and they can replicate independently, and they can primarily are carried from one generation to the other generation. They show the property of inheritance. And sometimes if you have natural plasmids, in some other occasions you can also artificially reconstruct a plasmid. That is, you can take maybe a small pieces of genes from different sort of an organisms, like a sort of a prokaryotic, like a eukaryotic cell or a prokaryotic cell, and later on, or from a fungal cell, and you can stitch them just like how you stitch pieces of cloth. You can stitch them and develop a sort of an artificial sort of a plasmid. So, an artificial plasmid is nothing but a reconstruct plasmid, which is done in vitro, that is in the lab, and later on placed into the sort of a bacteria. So, plasmids can be natural, or they can be an artificial, or it can be an artificial reconstructed. Now, if you look at the particular plasmid, in every particular plasmid, there is one region which is termed as an ORI. The meaning of the term ORI means it is the origin of replication. It has got a sequence of nucleotides which says wherein you primarily, it can make multiple copies of your sort of a plasmid. So, a plasmid, the extra chromosomal circular DNA, which has an ORI, can make multiple copies of itself. Making multiple copies of the plasmid is analogous to blowing lot of bubbles. So, you will have multiple copies of the sort of plasmids which keep on coming just like bubbles. A plasmid has got an ORI, and because of its ORI, it can make multiple copies. So, sometimes a plasmid can go to form only 10 copies. In some other species, it can make 1000 copies of itself. So, the larger the number of the copies, the larger the number of the copies of the gene you can get. So, there are some sort of plasmids which are termed as stringent. They make only 2 copies of itself. So, you can get only 2 copies of your recombinant gene. Whereas, there are other copies of the plasmids which are termed as relaxed. So, what are relaxed plasmids? Relaxed plasmids have got a very high copy number. They have a very high copy number. And thereby, the copies of the genes can easily be made more. So, it can make multiple copies of the gene can easily be known. Now, whenever you take any particular plasmid, again a few more characters of the plasmids is, the plasmid can easily be selected. There are some plasmids which can easily be selected by applying a sort of a fluorescent dye. And again, they also have selective markers. They have selective markers on the basis of which you can identify whether a cell has undergone or a molecule has undergone recombination, that is, has a foreign gene been inserted into the DNA. If a foreign gene has been inserted into a DNA by the technique of gene cloning, the cell which is getting formed is termed as a sort of a recombinant cell, and a recombinant cell will have the recombinant DNA, and the recombinant DNA is there within a particular cell because the plasmid has taken the sort of a DNA and it has shown it. Thereby, you can easily recognize or visualize or demarcate a particular recombinant cell from a non-recombinant cell. Common plasmids which are primarily studied are, we have got a type of plasmid which is called PBr322, which you will be reading shortly. PBr322 is a plasmid which has been discovered by 2 important scientists whose names are Bolivar and Rodriguez. and they have studied the strain called 322. And again, common plasmids which are primarily studied are plasmids called PUC19 and PUC101. PU primarily it's plasmid which is developed from the University of California. If you take plasmid PUC19 and PUC101 are a series of plasmids which have been developed from the University of California. Thus, we understand that before we really go into the details of the plasmids, you understand that plasmids are a boon for genetic engineering. Plasmids are natively present within the sort of a DNA. Plasmids are extra chromosomal elements. They are capable of inheritance. They are circular double-stranded molecules. They show a pattern of inheritance. They have an ORI or an origin of replication which is capable of having high copy number. Then again, they have selective markers and they primarily show all the sort of characters like whether they are easy to isolate, whether you can reintroduce a foreign gene. They could either be natural or they could be artificially constructed, and they are the carriers of the DNA and they can make multiple copies of the DNA. So, thereby now in plasmids, more specifically when you take the plasmids, we have one very important plasmid which is called PBr322. Now, PBr322 is a sort of a basic plasmid. It is just like the first series of rockets which get launched, or the first series of antibiotics which have been discovered. Similarly, when you take a sort of a basic plasmid, the basic plasmid which on the base of which entire molecular biology rests is termed as PBr322. It is just like maybe the first model of the car or the first model of the airplane whichever humans have ever seen. Now we will be specifically studying a very important basic cloning vector which is called PBr322. Now, PBr322 is a cloning vector which has been studied or constructed by Bolivar and Rodriguez. Bolivar and Rodriguez were two important scientists who primarily constructed the artificial plasmid which is termed as PBr322. So, it is an artificial construct, that is, it has been made in the lab. It has been studied by under in vitro. And when you look at PBr322, PBr322 is a circular molecule. It is primarily a circular molecule, and it is a small molecule, it is a small vector, it is a basic vector. It is a small and a basic vector, and it can easily uptake a sort of a DNA. The ease by means of which a foreign gene of interest can easily be loaded into this. So, it's an easy uptake plasmid. A plasmid can easily accept a sort of a foreign gene. Now, when you take PBr322, we understand it has a sort of a left flank and it has a right flank. The left flank is termed as AMPR and the right flank is termed as TTR. Now, what are AMPR and TTR? It is ampicillin and tetracycline resistant genes. It is a region which has got resistance to ampicillin, and this is another region which is resistant to tetracycline. So, AMPR and TTR genes are termed as gene selective, they are termed as selective markers. So, if you take the PBr322, it has got AMPR and TTR which are antibiotic resistant flanks. They are all popularly called as a sort of a left flank and the right flank of PBr322. So, these are important gene markers because if a foreign gene is taken by the plasmid and the plasmid gets into a sort of a bacteria and the bacterial cell gets transformed, then when we look at these particular ampicillin and tetracycline gene markers, from them we can exactly know how to select out cells. In a petri dish, when you have a medium, if there are non-transformed cells as well as there will be transformed cells, the transformed cells will have the sort of a foreign DNA, will have the alien DNA, will have your foreign gene of interest. So, these are termed as transformed cells, and all transformed cells will show the sort of properties of AMPR and TTR. Thereby, they are termed as gene markers. Coming back to PBr. When we take PBr, PBr has got one region which is termed as an ORI. Already you know ORI. ORI means the origin of replication. That is the place from which the plasmid can get replicated and it can go to form large number of copies. So, it is a plasmid which shows high copy number. And the plasmid is capable of taking a 5 to 10 kilobase pairs of DNA. It is capable of taking a sort of an insert, a foreign gene of insert, which is 5 kilobase pairs, can easily be loaded into the sort of a plasmid. There are different sort of plasmids which can take large molecules or small molecules or medium molecules. So, plasmids have got properties of uptake of DNA, they have selective markers, they show either high copy number or low copy numbers. Now, when you take PBR, it shows a high copy number, and again it is a low molecular weight plasmid. It is just like a sort of a fast shuttle. It is just like a sort of vector which moves from one bacteria to another bacteria, and it has got a low molecular weight. Imagine if supposing if it has got a very high molecular weight, for it to travel from one bacteria to another bacteria, it will take a lot of time. So, it is a low molecular weight plasmid which can take in your sort of foreign gene of interest, and most thoroughly studied plasmid by Bolivar and Rotritz. So, it has got an ORI, it has got gene markers, and if you look, it has got a gene which is termed as ROP. Now, ROP is a gene which codes for proteins in replication. It codes for proteins in replication, and it again, the entire plasmid, which is a circular artificial reconstruct, is recognized by a battery of enzymes, a battery of restriction enzymes. This is the region where PST1 will cut the sort of a plasmid. This is the region where PVU will cut the sort of plasmid. This is the region where ECOR1 and CLA1 as well as HIN3 and BAMH1 and SALH1 and PST1 will cut the sort of a plasmids. So, a plasmid primarily is similar to that of a bangle, which can be broken into different sort of pieces or which can be cleaved by means of a battery of restriction enzymes. Now, these restriction enzymes recognize, like if we take ECOR1, it recognizes the sequence over here. So, when it recognizes the sequence over here, you can load your sort of a gene of interest. Your gene of interest can be loaded over here. And thereby multiple copies of the same sort of a gene can be made because the plasmid has got a very high copy number. So, it is very, very important for us to understand that plasmids are basic tools in genetic engineering, and a good plasmid will show easy uptake, it should have high copy number, it should be small, and it should have a low molecular weight, and it should be well thoroughly studied, and again it should have an ORI, and it should have unique restriction sites. What are the unique restriction sites in the case of PBR322? We have got PST1, PVU2, we have got ECOR1, again CLA1, then again we have got HIND3, BAMH1 and also SAL1. So, we understand it has got 7 important restriction sites. It has got an ORI, it has got 2 gene markers, it has got ROP, it has got ROP gene, and it is an artificially reconstructed plasmid. So, whenever we talk in terms of plasmids have got a lot of advantages in genetic engineering. The advantages which the plasmid has got with relation to genetic engineering is, first important thing is they show different sort of properties, such as it should be small, it should be an in vitro construct, and it should have a high copy number, it should be easily be taken up, and again it should have at least minimum of 2 to 3 gene markers, and it should have unique restriction sequences, and it should have been thoroughly studied, and it should be used as a sort of a tool in cloning experiments. A modification of PBR plasmid is termed as a sort of a cosmid. It is just like we have different series, like if you take plasmids, from a plasmid you have the development, the next generation of vector is termed as a cosmid. So, what is a cosmid? A cosmid has been developed by a scientist whose name is Cohen in 1978. A cosmid is a plasmid with a cos site. A cos site is a cohesive site. A cohesive site is derived from another sort of a bacteriophage which is termed as a sort of a lambdaphage. A lambdaphage is a linear bacteriophage, and it has got a region which is called a cos site. So, this cos site has been taken and attached to a sort of a plasmid, and a plasmid with a cos site is termed as a sort of a cosmid. So, we have different sort of a constructs of vectors like a sort of a plasmid or a cosmid. So, it is very important that what are the basic properties of plasmids is it should have an ORI, then it should have selective markers, and selective markers, the vector requires a selective marker for identification of transformants and for elimination of non-transformants. The selective marker will enable to know whether the transformants are growing and the genes, and it should have maybe most of the transformants will have a sort of a gene for antibiotic resistance. So, now when we take a particular sort of a vector, again a cloning vector should have cloning sites. The cloning site should be a single recognition site, or here you got two important recognition sites. You got an AMPR and a TETR, into which you got the different sort of a restriction enzymes coming in. So, commonly which is recognized, the recognition site of common restriction enzymes should easily be known in a sort of a plasmid before it is used for genetic engineering. And again it should have a low molecular weight. That is a very important characteristic feature of any particular sort of a plasmid. Now, in addition to the plasmid, so we have studied primarily about restriction enzymes. Next, we have also studied primarily about plasmids. The third important component of a tool is the presence of a sort of a competent host. We had seen the tools in plant biotechnology as A, we had completed restriction enzymes. Next, B, we had finished cloning vectors. Now, what again are the sort of a tools? Among the tools, a very important thing is the presence of a competent host. A competent bacterial cell is a competent host. A competent host is one which can uptake the sort of a recombinant DNA. How can you exactly know whether the bacterial cell is taking the sort of a recombinant DNA? What is its capacity? What is its capacity to accept the sort of a recombinant DNA? That is termed as competent host. Now, in order that a host be competent, it is very important that it should accept the recombinant DNA, and one method of how it can accept the DNA is if we add calcium divalence. When we add calcium divalence to the host cell, the cell walls, the pore size increases. Once the pore size increases, it is easy for the foreign DNA to be taken in by the bacterial cell. So, it improves the efficiency of competence. So, efficiency of competence can be by adding calcium divalent ions on the sort of a membrane. Thereby, you will find that a bacterial cell, after it becomes competent, we say it has taken up the sort of a foreign DNA of interest or the foreign gene of interest, and the host cell is supposed to be genetically transformed. So, one method for the competent cell is by means of using of calcium divalent ions. In addition to these particular competent host cells, there are also methods by means of which we can do vectorless transfer of your foreign DNA. The vectorless transfer of the foreign gene or the foreign DNA is possible by the technique of electroporation. Now, what exactly is electroporation? Electroporation is a vectorless gene transfer technique by the use of electrical pulse. So, if we take plant cells and we apply a low voltage for a longer duration, or we apply a second situation, a high voltage for a shorter duration, we increase the pore membrane space. When we increase the pore membrane space, the possibility of taking a foreign DNA is 10x times than when compared to that of a vector. So, modern techniques of how you do an uptake of a DNA is also by the method of electroporation. In addition to this, like if you take electroporation as well as use of a competent cell, what are the sort of an advantages or the applications of this particular method? It's easy to exactly know how a drug is delivered, how you can do drug delivery into a sort of a plant cell. Which sort of a drugs, when you put in a sort of a drug or you want to put in a sort of a gene which is responsible for the development of a drug, it's easy to probe that. So, you can use it as a sort of a molecular probe. Like you can have a sort of a gene or a sort of an enzyme which goes and exactly you can know the pathway of how the particular gene is moving. And you can also study a sort of a DNA insert. Now, in addition to electroporation, another method is which is very popular in plant molecular biology is by means of the particle or the bioelastic gun method. In the particle or the bioelastic gun method, we have a gene gun. A gene gun is a sort of a instrument which primarily wherein you have the DNA, and the DNA is coated by means of gold or tungsten particles. The DNA, the foreign DNA of interest, is isolated and it is coated by gold or tungsten particles, and it is shot into the plant tissues like a sort of a ballistic missile, and directly the DNA can be integrated or injected into the sort of a palisade tissue or into the sort of a chloroplast. So, it is termed as a particle or a ballistic gun, and this is a direct method of genetic transformation, and the process is termed as the genetic transformation of plants. So, genetic transformation of plants, especially in the case of plants, the most common method is the gene gun method or the particle or the ballistic gun method. In addition to this, whenever we want to take up a particular DNA, we also have chemical methods. The chemical method involves the use of a chemical which is termed as a polyethylene glycol, PEG. When we use polyethylene glycol, we can primarily enable protoplasts to do the sort of a naked protoplast fusion, and when there is naked protoplast fusion, there is a sort of a direct DNA uptake. So, direct DNA uptake by means of chemical methods is by means of polyethylene glycol. So, we understand primarily in the tools a competent host primarily, and different methods by means of which a foreign DNA can be upgraded are primarily put in the form of a sort of a map over here. We have electroporation as one method, ballistic particle gun method, and also polyethylene glycol as one of the sort of a methods. So, this way we primarily finish off on the sort of a tools which are used in plant biotechnology.